like to thank the organisers very much for giving me the opportunity to present this work, which is the result of a large collaboration between individuals from 5 different countries. So to begin with, I'd like to draw your focus to the hypothalamic-pituitary-thyroid axis in the context of congenital hypothyroidism. Primary congenital hypothyroidism has a prevalence of around 1 in 3,000, and it arises due to a primary structural or biosynthetic defect in the thyroid gland itself. It's associated with decreased levels of circulating thyroid hormones, and because negative feedback at the level of the hypothalamus and pituitary is reduced, TSH is elevated in this condition. In contrast, isolated central congenital hypothyroidism is a much rarer entity, with an incidence of around 1 in 40,000. It occurs due to a primary defect in the hypothalamus or pituitary. And in this condition, circulating levels of thyroid hormone are decreased, but they're associated with inappropriately normal or subnormal TSH levels. Untreated congenital hypothyroidism is associated with profound neurodevelopmental delay, and therefore it's screened for in developed countries, usually with a blood test within a week following birth. In common with most other European countries, the UKCH screening programme relies solely on measurement of elevated TSH on a filter paper blood spot sample, and because of this it will detect primary congenital hypothyroidism but not central CH. In other countries such as the Netherlands, T4 is included in the CH screening protocol, and therefore both these entities will be detected. I'd like to present 2 index families in which there were affected cases with isolated central hypothyroidism. Family A was from the Netherlands, and in this family, the proband and his affected male cousin were both diagnosed with central hypothyroidism on neonatal screening. Family B was from the UK, and in this family, there were 2 male siblings who were found to have central hypothyroidism fairly early in the neonatal period when they presented with prolonged neonatal jaundice. To date, the only known genetic causes of isolated central hypothyroidism are mutations in the TSH beta subunit and TRH receptor genes. We screened both these genes in our kindreds, but we did not find any mutations, and we went on to use whole exome sequencing. This enabled us to identify mutations in the immunoglobulin superfamily member 1 gene, IGSF1, in affected individuals in both families. Now, IGSF1 is located on the X chromosome, and when we carried out family screening, we were able to identify hitherto undiagnosed males with central hypothyroidism who harboured mutations in the same gene. We went on to screen IGSF1 in other unrelated families in which there were males with central hypothyroidism, and we've now identified 8 distinct mutations and 2 whole gene deletions in 11 unrelated families. So what's known about IGSF1? Well, it's on the X chromosome, it's highly expressed in the pituitary from early on in development, and it's also expressed in adult testis. It's a membrane glycoprotein, but its precise function remains unknown. The protein undergoes co-translational proteolysis such that eventually the carboxy terminal portion of the molecule traffics to the cell membrane, and the majority of the molecule is located extracellularly with a short cytoplasmic domain. All the mutations that we've identified to date affect this carboxy terminal region, and they include 4 in-frame mutations and 4 premature truncations, together with the whole gene deletions that affect IGSF1 alone. So we went on to ask the question as to how these mutations might affect IGSF1 function. All the truncation mutants would be predicted to terminate the protein before this transmembrane domain, so you might predict that these mutants would not be expressed at the plasma membrane. And we tested this by overexpressing wild-type and mutant protein in HEC293 cells, and then using an antibody specific for the extracellular domain of the molecule to detect plasma membrane expression. And you can see that although both wild-type and mutant protein are clearly expressed intracellularly in permeabilized cells, in non-permeabilized cells, none of the new truncation mutants are detected at the plasma membrane, in contrast to wild-type protein, which is abundantly expressed. We knew from similar experiments with the in-frame mutants that we could detect some plasma membrane expression of these mutants, so we went on to use a cell biotinylation assay in order to investigate plasma membrane trafficking and expression of the in-frame mutants in more detail. So you can see that wild-type IGSF1 migrates as two distinct glycoforms, an immature and a mature form, but it's only this mature form that can be efficiently trafficked to and expressed at the plasma membrane. Although the in-frame mutants are all clearly expressed within the cells, they all migrate as the immature glycoform, and this explains why there's negligible plasma membrane trafficking and expression of these different mutants. So all the mutants that we've identified so far affect plasma membrane trafficking and expression of IGSF1. We then went on to investigate the clinical phenotype in our patients. We've now identified 26 affected males in our 11 different families, and they all have central hypothyroidism. They all have subnormal T4 levels with an inappropriately normal TSH. But although the central hypothyroidism is a universal phenotype, you can see that there's quite marked phenotypic variability. So some individuals have quite profoundly decreased T4 levels, whereas in others they're only just below the lower limit of the normal range. 8 individuals had TRH tests prior to commencement of thyroxine treatment. In infants, the TSH peak tends to be blunted, whereas in adults it tends to fall within the normal range. And we also found that just over 50% of our cases also had prolactin deficiency. We also assessed pubertal development in those patients for whom data was available, and we found that this followed a characteristic pattern. Testicular development was initially normal. Then the pubertal growth spurt and testosterone surge tended to be delayed, but in late adolescence, even if testosterone levels were still low, testicular size tended to increase, and all affected adults had true macroorchidism, with some having testicular volumes in excess of 70 mils. Cases could be divided into 3 different subgroups, depending at the age at which their central hypothyroidism was diagnosed. So a subgroup were diagnosed in the neonatal period, and this was either because they lived in countries where the neonatal screening program picked these up, or because they presented with symptoms suggestive of neonatal hypothyroidism. A further subgroup were diagnosed in childhood, usually because they presented with symptoms of childhood hypothyroidism. But I'd like to draw your attention to this large subgroup here, who were relatively asymptomatic and only diagnosed in later life following the identification of a younger affected relative and subsequent family screening. Within that subgroup, all had decreased levels of T4. Some had clear biochemical evidence of hypothyroidism and tissue evidence of hypothyroidism as well. So for example, this individual has clearly subnormal T4 levels, he has a raised CK, a subnormal sleeping heart rate, and a markedly subnormal basal metabolic rate. But even for those patients who were more mildly affected, they still had overt hypothyroidism, albeit mild. And there's emerging data that even subclinical hypothyroidism can be associated with adverse cardiovascular function and mortality, which responds to thyroxine treatment. And I think this really adds weight to the importance of genetic ascertainment in this syndrome, so that even these relatively mildly affected individuals are identified early on so that they can maximally benefit from the effects of thyroxine replacement. We went on to use a mutant mouse model to try and investigate the mechanism underlying the central hypothyroidism in our patients. So IGSF1 delta exon 1 mice lack exon 1 of IGSF1, and the males express virtually no IGSF1 protein at the plasma membrane. They are centrally hypothyroid, they have decreased serum T3 levels and decreased serum TSH levels, but like the humans, there's quite marked phenotypic variability. And in keeping with biochemical hypothyroidism, they also have elevated levels of plasma cholesterol. These mice have normal levels of TSH beta messenger RNA in the pituitary, but levels of TSH protein are decreased, and the serum TSH response to a TRH challenge is also blunted in these mice. Given the known importance of TRH in driving TSH protein maturation and release, we investigated TRH signaling in the mice, and we found that in the hypothalamus, levels of TRH messenger RNA were increased, but in the pituitary, levels of TRH receptor were decreased. So whilst we do not by any means have a completely comprehensive mechanism to explain the central hypothyroidism in our patients, we believe that in IGSF1 deficiency, TRH signaling is impaired, and this is at least in part due to decreased TRH receptor expression in the pituitary. This results in decreased TSH maturation and release, with subsequent decreased circulating levels of thyroid hormone. So in summary, IGSF1 mutations cause a novel X-linked syndrome of central hypothyroidism, adult testicular enlargement, and prolactin deficiency. Defects in TRH receptor expression and TRH signaling may underlie the central hypothyroidism, and the fact that more than 50% of patients have additional prolactin deficiency suggests that the TRH signaling deficit may not be limited to thyrotropes. Adult macroorchidism is a consistent feature in this syndrome, but the mechanisms for this remain unresolved. And the detection of biochemically affected but asymptomatic cases highlights the importance of genetic ascertainment in this condition. And it remains to thank my collaborators both from the UK, the Netherlands and Italy, and also to acknowledge the chief UK funding bodies, the Wellcome Trust, the NIH, BRC. Thank you for your attention.